Hi everyone, welcome back to the channel. This is Dr. Asin, and today's topic is cell. This is chapter number 3 in Orange Book, a tour of the cell or cell. So let's get started. Okay, so cell chapter starts with microscopes because biologists use microscopes and some other tools of maybe biochemistry or cell biology to study cells. And we will see the reason for that. We have two student learning outcomes in this chapter, which we cover cell structure and organization and understanding of structure in order to understand the function. And also there is a second part, which is membrane transport. We will focus on membranes and how actually transport work on the membranes and mechanisms. So, of course, we divide the cells into groups, eukaryotic versus prokaryotic. Prokaryotic cells appear first and much simpler cells, and eukaryotic cells came later and much complex cells. Eukaryotic cells have internal membranes and they compartmentalize their functions. And they also have genetic instructions housed in a nucleus, specifically and carried out by the ribosomes. And then we will talk about endomembrane system. So, and then we will talk about some specific organelles like chloroplast or mitochondria. So, okay, so let's get started. The chapter starts with microscopes. The microscope, they go back to 1665, Robert Hooke, and first microscope designing using glass lenses. Today we know those microscopes as light microscopes. We use it in everyday life, which basically for regular cell studying cells. So magnification and resolving power important in the microscopes. So we will see examples. So there is so again, this started with 1665 by Robert Hooke. So since then, and last 350 something years, so now we have actually cell theory. According to cell theory, all living things are composed of cells, and cells come from pre-existing cells. All cells also contain hereditary information like DNA. So today we have also much more complex microscopes, like they are called electron microscopes, EMs. They can actually they can be used in to see much smaller things under electron microscopes with much higher resolution, like scanning electron microscope SEMs for cell surfaces, and TEM, which is transmission electron microscopes, studying internal structures. Like you see pictures here in this slide, a scanning electron microscope and transmission electron microscope and light microscope of the same organism. Okay, so this is just an electron microscope here is on the list. You can see it can go up to the 100,000 times of magnification, which light microscopes could maybe go only 400 maybe. So this chart is showing you how small the things are in the cell, so that they require microscopes, like plant animal cell itself, nucleus, some bacteria, mitochondria can be seen under light microscope, but viruses, ribosomes, proteins, lipids, atoms, they require electron microscope. Okay, so again, we have two groups of cells, prokaryotic and eukaryotic. Prokaryotics are like bacteria and archaea, all the phylums, and eukaryotics, protista, animal, plant, fungi. So all cells have a plasma membrane. Most cells have wall, but not animal cells. All cells have cytosol. All cells have chromosomes. All cells have ribosomes, which are the protein factories. And eukaryotic cells appear around 2.1 billion years ago. Eukaryotes, so we will spend more time on eukaryotes. We have animal and plant cell in eukaryotes. Basically, they have 16 to 18 number of organelles, but there is also prokaryotes. You can see here on this slide. There is plasma membrane, and some have cell wall, and there is ribosome, and there is some DNA inside. So a very important slide, which is animal cell versus plant cell. Animal cells have about 16 organelles. Plant cells have about 18 organelles. They have a lot of organelles in common, except two organelles. Maybe animal has, plant don't have, like centrosome and lysosome. And plant have, animal don't have, cell wall, chloroplast, central vacuole, amyloplast, and other organelles are actually shared by both animal and plant. And in many of these you see in this picture. So a plasma membrane is one organelle that actually all cells have. It doesn't matter what kind of cell it is. It separates living from unliving. Anything inside PM is living. Anything outside is non-living, and they are made of phospholipids, bilayer, and there is this sandwich model you can see in this picture. You can see the sandwich-like structure, which actually the purple items you see here, they are the proteins that they're going through, or they are on the surface. So in animal cells, there is no wall, but sometimes there is extracellular matrix, which is basically outside of plasma membrane. Nucleus, obviously, very important in animal and plant cells. Most DNA is located in nucleus, inside nucleolus, and there is also proteins and chromatin and other stuff. So you can see here chromosomes inside nucleus, which are contained in DNA, which contains the genes, nucleotides, and the double helix DNA structure. Ribosomes, very, very important organelle. Again, all cells have them, and they can be, they are the protein factories of the cell. They can be attached to ER, or actually they could be separate and just floating around without attaching to the ER. So there is this whole information flow from DNA to mRNA and inside nucleus, and then the mRNA leaving nucleus, finding a ribosome, and then getting inside ribosome and producing actually polypeptide proteins. So there are in animal cell there are 16 organelles, but a few of them are actually directly linked. They are called endomembrane system, and they work together very closely. Some of them, again, nucleus and Golgi is another one, which is basically, okay, sorry, before Golgi, there is ER, endoplasmic reticulum, which look like in the picture here. There is rough ER, is the ER that has ribosomes on it, you can see, or smooth ER, which has no ribosome in it. So rough ER produces proteins, obviously, because of ribosomes. Smooth ER is also important. It can actually produce lipids, steroids, and some other molecules. Golgi is this pita bread-like structure you see on top of each other, which is a receiving side, and then trans side with shipping side on the opposite direction. Okay. And it's like a post office, receives stuff and then prepares them and ships them out. Lysosome is an animal cell specific organelle, which has a lot of digestive enzymes. Most plant cells don't have it. They can break down large molecules like proteins, polysaccharides. And this is just showing all these three. They can actually destroy harmful bacteria or some damaged organelles. You can see here lysosomes and this cell structure. Vacuoles, we see them mostly in plant cell. the plant cell specific, especially the central vacuole. So they might be storing nutrients, some water, and pigments. As you might know, plants have a lot of pigments, green, orange, yellow, red, brown, purple, or some other colors. So the plants are very colorful. Chloroplast is one of those plastids, which is an organelle that actually very important in plant and algae cells. Basically, it has the pigment of chlorophyll, which is we see as green color, like chlorophyll B. Chloroplast is made of stroma inside. There is these thylakoid membranes inside also, which are all plurally called granum. So, but chloroplast important because obviously they carry out the most important reaction on biosphere, which is photosynthesis. So another organelle kind of linked directly to the chloroplast is mitochondria, which is you can see they both are very independent organelles. Mitochondria also has a lot of stuff like the chloroplast, like its own DNA, ATP, energy, matrix. You can see and crystal, these foldings, that parts of the mitochondria. Again, mitochondria has its own DNA, like as chloroplast has its own DNA. Outside of nucleus, two other places, mitochondria and chloroplast, they have their own genome and DNA and genes, it means, or chromosomes. So cytoskeleton is basically we see in both animal and plant, like a networking thing that which basically has microtubules and intermediate filaments. That's actually, let's see, they give like you see in this picture, they provide shape to the plant cell or cell. It's very dynamic. They might have a flagella connected to that, helping them maybe some movement, cilia, another thing like little hair-like structures. So these are some general like big organelles. Make sure that you actually keep reading from the chapter 3, all the other organelles and their functions. Plant versus animal versus bacteria, plastids we talked about. And the next thing is talk a little bit about membranes specifically and transport. So plasma membrane, PM, is the most important membrane. It's called selectively permeable. That means basically it lets only specific thing cross easily than some other things. So I already mentioned plasma membrane is made of 2 layers and it looks like a sandwich model. So here is a real cool picture of this liquid mosaic model, it's called. This wall-like structure you see here is PM, plasma membrane, which have all these proteins on it in purple. They can be peripheral on the surface or integral, which is crossing the membrane. They have glycoproteins, carbohydrates, glycolipids, and cholesterol. Very important here, you can see. Okay, so, and outside, you can see extracellular matrix. Inside is basically inside the cell. Okay, like microfilaments and other structures. So steroids I just mentioned is inside the plasma membrane. So temperature can affect how actually how membranes behave. So cool temperatures, they are more fluid. Well, so proteins, very important, right? So we already learned about proteins in last chapter. So we know one of the major function of protein is transport, right? So here where they are going to locate it, either on the surface or inside the membrane to help this transport. These are also called the ones going through membrane called transmembrane proteins. So some other functions of proteins like transport, enzyme, signaling, cell recognition, and attaching to the cytoskeleton. So some other 6 major functions of proteins. So carbohydrates and lipids are there also in the plasma membrane, but we are going to more look at the bilayer of the plasma membrane. So there is hydrophobic part, which is more inside, and hydrophilic part, which is outside, right? So there are group of proteins called transport proteins. One example, aquaporin, which basically a channel for water, transporting water, aquaporin. Transport can be in 2 groups, passive, which is basically like a natural diffusion, which is free. It goes from high to low across the plasma membrane. There must be a gradient, high to low, in order diffusion works. It's like I am spraying a perfume here, and then it just slowly move from here to the end of the room, basically diffusing, right? So osmosis is a special diffusion of water, which is same idea. It goes from more wet, more water, to the less water, right? So in plants, this is very, very important, because plant cells cannot really store water. So like you see here, animal versus plant. And a normal, which will be isotonic, which means inside and outside water is similar, same. Hypertonic is there is less water inside. If it is animal, it's called shrivel. If it is plant, this is called plasmolyzed. So plants don't have enough water. If you give them a lot of water, they become turgid. If it is animal cell, they are called lysed. So isotonic is same. Hypertonic solution is there is less water inside cell. There is more. There is, and this situation is hypertonic. Okay, so diffusion, it can be just simple diffusion or facilitated diffusion, like using a channel like this, again from high to low, like ion channels. And another transport is active transport. Active transport is basically is not free. It uses a energy, like you see here. This is passive transport, diffusion, facilitated diffusion. Active transport, you can see ATP has to involve. Then direction doesn't matter. It can go uphill, no problem, because it uses energy. So it requires energy. A good example is sodium-potassium pump. You can see on the right side of this slide, it takes the potassiums. Sorry, it starts here. Takes the sodiums, goes to, when it goes the other side, it relays the potassiums and get sodiums, relays the sodiums, and then take potassium and brings potassium. So it uses transport both sodium and potassium, right? So there is another item that's called membrane potential. It's not just concentration chemical, but there is an electrical component too. So there's a charge outside cell and inside cell. Cell is considered negatively charged. So there is an electrochemical gradient. Obviously, anything try to move in which is negatively charged, like chlorine, is gonna have more difficulty, because there's cell is negative, chlorine is negative, is not gonna attack each other. So the cells uses both electrical and chemical force, which is also called altogether electrochemical gradient. So sodium potassium pump is a good example. We can see here in another pump, which is sucrose co-transporter, sucrose proton co-transporter. You can see sucrose moves. So first, the hydrogen ions actually move outside from the cell membrane using ATP, and they actually create a momentum here, and then help moving inside through the second transporter here. And then while doing that, that transporter takes sucrose in too. So it's like a coupled transport. There is also bulk transport for larger items that can be phagocytosis, pinocytosis, or some receptor-based endocytosis. Exocytosis is moving things out, large items. Endocytosis is moving them in. So there is a picture here showing how large items can be moved out or in, like bulk items, using bulk transport, again through plasma membrane. So in your book, there is obviously, this is chapter 3. There is a little summary here in the PowerPoint slides. Make sure you study these, complete the quiz, and I will see you in next video. Thanks for watching. Please feel free to comment, subscribe, and like. Happy studying.